I sent them an email. I'm not sure if we need.
I'm gonna start. Okay, Brian took care of it. Thanks. All right. Well, hello everybody. Good morning and welcome to the Department of Psychiatry and Behavioral Sciences Grand Rounds. Today, it's my pleasure to introduce Dr. Nicole Lawson. She is a clinical pharmacist who works at Woodridge Psychiatric Hospital as the pharmacist in charge there. And Dr. Lawson completed her training. First, she went to Wytheville Community College for her Associates of Science in Dental Hygiene, worked as a dental hygienist for a few years before going to pharmacy school at the University of Appalachia College of Pharmacy. She graduated there in May 2008, and since then has been in various different pharmacy roles and has currently serving at Woodridge, where she works very closely with the residents in any of our pharmacy needs. So without any further ado, it's my pleasure to introduce Dr. Lawson. Thank you.
Oh, thank you, David. Good morning, everybody. I just want to start by thanking you all for inviting me to come and speak today and be a part of Grand Rounds. As David said, I graduated from, it was the University of Appalachia College of Pharmacy at the time, and now it's just ACP, with my doctorate in pharmacy in 2008. I worked full-time at Walgreens and part-time at a small community hospital right after graduation. And since then, I've kind of dabbled in several different areas of pharmacy, independent pharmacy, some manufacturing, some sterile compounding, pain management. I teach a pharm tech class at Southwest Virginia Community College. But I really was never satisfied until about 4 years ago when I started hospital pharmacy full time. And then I found my focus in psychiatrics. I started studying the pharmacogenomics several years ago, and I'm really excited about where the particular research is headed. So today we're going to take a look at pharmacogenomics, using pharmacogenomics to advance medication use. I'm going to take me off of here. Can you see the screen okay, David? Everything good?
We can, and we can still see the screen. If you want to turn your camera back on, we could still see your PowerPoint.
Okay, I'm good.
Okay.
I'm okay with that. Okay. So we will start off translating science into practice. This is really what we're doing with the study of pharmacogenomics, and we're trying to take the information that's been discovered and translate it into something that we can use clinically to increase patient care and to make it better. As a clinical pharmacist, I'm always trying to think outside the box. So in my opinion, the study of pharmacogenomics, as it grows, it can have really endless possibilities. Today, we're going to just touch the surface of the study of it so we can look at how that it can possibly advance medication use and patient care. Some key abbreviations that you will probably need to know. Pharmacogenomics is always noted as PGx when you're reading studies and throughout this presentation. Also CPIC, and we'll learn more about that database in a couple of slides, but it's the Clinical Pharmacogenetics Implementation Consortium, and that's always abbreviated. So all in all, what is pharmacogenomics? It's the science of identifying and validating genomic variants influencing drug response and implementing strategies to such genomic information to inform treatment decisions. Aside from cancer and infectious disease, and we're not really going to touch on those two disciplines, they are actually a little bit different. Somatic acquired genetic variations must be considered in cancer, and then variation also impacts pathogens, antimicrobial sensitivity when we're looking at infectious disease. So we're really not going to cover anything in those two today, and we're going to focus more on the psychiatrics. So the bottom line is the study of how genes affect a person's response to drugs. And the goal is to develop effective and safe medications and doses based on a person's genetic makeup. So a long time ago, pharmacogenetics dates actually back to more than 2,000 years ago to observations by the Pythagoras. In the 1950s, some enzyme polymorphisms were discovered, but newly uncovered information was considered too obscure for that time period. So it's kind of too risque for that time period. But it wasn't until the 1970s when the discovery of CYP2D6 polymorphism and the effect it had on drugs toxicity would lead to individuals examining the new term and the effect it had on a drug's pharmacokinetics. The goal of the research really is to move away from a one-size-fits-all dosing. In 1984, the US government began a project called the Human Genome Project, and planning started, and the government formally launched it in 1990. It was declared complete on April 14, 2003. It was kind of an international research project that tried to determine the base pairs that make up human DNA and identify and map all the genes in the human genome. So where will pharmacogenomics take us? Patients are always interested in their pharmacogenomic data. They're always interested in what medications go best for them. There are numerous genes for which polymorphisms have been identified that are associated with variable drug response. And this could actually be at the drug's metabolizing enzyme, at the transporter, or the receptor level. The translation of pharmacogenetics into personalized medicine, though, has several barriers. And these barriers actually include the clinical relevance, the environmental genetic interactions, the cost, and education. And actually, it's education, education, education. And as we learn, as we go on through the slides, that's our biggest barrier is I think as the information comes to us, we just need to make sure that we are educating ourselves and keeping up to date on what's going on with the study so that we can further the advancement of it. So medications are a leading source of patient harm. So as I was looking through some of the studies, there is a study done in North Carolina hospitals that say that medications were concluded to be the leading cause of patient harm. And this isn't really talking about medication errors like in administration, but about a lot of people being harmed by just prescription drugs that they're taking, but they're taking it as prescribed. Sometimes the medications themselves just aren't correct for that particular patient. So therefore, by finding out the best medication for a patient to start and the dose that works best for them, we always, in pharmacy, that's our motto, start low, go slow, is a proactive patient safety strategy also. So let's move on to testing. Testing is usually done on blood or saliva. There are three major reasons that we would do pharmacogenomic testing. The effectiveness of a medication that we're testing to see, the dosage of a medication, or potential for a serious side effect of a medication. So the question is always, who should we test? Pharmacogenomic testing is only used to find out a person's response to a specific medicine. So the test can occur before and while a patient is on treatment, and it can help a provider to figure out the right dose. So some tests, such as the ones for certain drugs like for epilepsy and HIV, can show whether you are at risk for a life-threatening side effect also. So that helps the provider to find alternate treatment so that the treatment that may cause the problem is not even started. The cost of the test are decreasing, and some insurances are actually covering the cost because it's overall lowering healthcare costs. By number one, it avoids the trial and error, and it it actually decreases medication overall cost. Past studies, sometimes they've shown that 38% of Americans do not metabolize antidepressants properly, and 50% of Americans are poor metabolite, like have poor metabolites to arthritic medicines also. So there are only a limited number of examples. As you can see, whoops, let me go to the next one. There you go. I just put up a couple, like there are tons more than this, but I just put a couple on this slide that you can see. I would like to add that morphine also affects the 2D6 gene. And I just wanted to put that one in there too, because this one only put the OPRM1 on there. An example of an adverse event that can be avoided is codeine, oxycodone, hydrocodone, and similar drugs have a warning against prescribing them to patients who are rapid metabolizers. And this is because the drugs converted to morphine and ultra rapid metabolizers will conduct them from morphine to a greater extent. So it can lead to actually a fatal adverse event. I'm going to put everybody on pause 1 second. Hey, hang on just a second. I'm going to take just 2 minutes if everybody will hang on. I'm having a little bit of technical issues and I will be right back. I did like the music though. That was nice. Thanks, David. I can tell by the smile that might have been you. Nope. So we're gonna move on to pharmacogenetic testing in psychiatry. So there's been a recent review on November the 10th, 2020 that came out, and it was published in the Journal of Pharmacopsychiatry. The review provides a summary of recent developments that clinicians should know when considering pharmacogenetic testing for behavioral health patients. This is provided in sections of pharmacogenetic mechanisms, pharmacogenetic evidence, and guidelines for psychiatry and pharmacogenetic testing in psychiatry. This is a really, really good read if you want to go back and find that. I didn't put it in here or anything. I didn't really reference much in it, but it's actually a really good read to pull up, and it gives a lot of good information. So here we go. 1, 2, 3, action. 95% of experts in pharmacogenomics that were asked their top 3 challenges to implementing pharmacogenetic testing in the clinic responded that the process required to translate genetic information into clinical actions was a problem. The genotype test interpretation was a problem, and providing recommendations for selecting the drug gene pairs to implement was also a problem. So again, who should we test? Number 1, patients taking a large number of medications with the potential for serious adverse effects. Number 2, patients taking medications with a high risk of drug interactions. Number 3, when patients don't really respond to a drug treatment and the options are very limited, whether it be from known allergies or whatever their limitations may be. And number 4, with patients with an unexpected pattern of side effects to medications. Those are really our 4 patients who, in a clinical setting, that we would want to to kind of see before we even begin treatment of what would work best for them. Pharmacists are embracing pharmacogenomics as an opportunity to improve patient care and professional responsibility. And there are suggested standard operational procedures and models that are out there, but most are actually inpatient focused at this time. However, some outpatient models exist and are often dedicated to clinics run by pharmacists. Healthcare systems are often devoting an entire position to the new research that's coming out and to the pharmacogenetic implementation. And there's a lot of literature out there in databases. The problem is trying to find where it's at. Scientists had the foresight to create agencies to monitor the pharmacogenomic literature. Upon completion of the sequencing of the human genome is when that began. And they have two sites that are really good. They initiated an open access line and a database so that clinicians and healthcare professions could access all of that with the CPIC. And I'm going to move on to the next one to discuss what that CPIC is. It's an example of a valuable resource that guides the use of genetic information when available. It's an international consortium of individual volunteers and a small dedicated staff who are interested in facilitating the use of pharmacogenetic tests for patient care. And the group began as a shared project between PharmGKB, and this research group was actually managed at Stanford University since 2001. And Pharmacogenomics Research Network, which is PGRN, in 2009. A big barrier of why we haven't implemented pharmacogenetic testing in clinics everywhere is the difficulty in translating the genetic laboratory test results into something that we can actually use in the clinic. The goal of this whole community of databases and information coming in is to address the barrier by creating and implementing clinical practice guidelines. Financial support actually for this usually comes from the NIH also for this consortium. This slide actually, I just put in there to reiterate the validity of this site. They are indexed in PubMed as clinical guidelines endorsed by the ASHP and ASCPT and referenced in ClinGen and PharmGKB. And as of December 2018, they are also supported by the College of American Pathologists, which also goes back to, we're not going to touch much on the cancer stuff, but that actually, or even with the pathology, but it actually is endorsed by a lot of different groups now. CPIC's mission is to evaluate all evidence-based, peer-reviewed publications. And again, I just want to say peer-reviewed. As with anything else, it needs to be a peer-reviewed site. Take those and create and maintain the database with the newest gene drug guidelines. The way that CPIC works, it assigns an evidence level of either A, B, C, or D. A and B have actionable prescribing based on genetics, and C and D are not considered to have any adequate evidence to support. And this can be a useful starting point in deciding whether that clinical genetic testing of that gene for that drug can be justified. For example, in 2015, CPIC published a comprehensive guideline summarizing the evidence base for clinical decision-making when prescribing SSRIs affected by the genotypes CYP2DC and 2C19. Just to mention a second organization too, I know I said it before, but I didn't tell you what it was. It's the International Society of Psychiatry Genetics. It maintains an active an evidence-based website. It continually updates also new information and any kind of research development. One thing with this group, like with the second organization, they last came out with their recent statements about HLA-A and HLA-B testing prior to the use of oxcarbazepine and carbamazepine. They actually said that they studied both of those things, and then they're most likely to be beneficial for patients who've experienced an inadequate response or adverse reaction to any kind of antidepressant or antipsychotic trial. So that was their conclusion on that. And those both go back to the 2D6 and the 2C19 genome. So next is a quick overview of just the pharmacogenetic dissemination. I feel that we're barely scraping the surface of eventually what is going to be really big in healthcare with the use of pharmacogenomics. Just by looking at this slide, you can see that there's so many different things that are being reached and so many different places that are not being reached. So this is definitely going to be ongoing for many years. I'm going to take just a second. I'm going to get a drink. So to get to the point where we can grow and have all this research and development is teamwork, and it's going to be teamwork between our pharmacists and our physicians. So the future success of PGx relies on the collaboration of pharmacists and physicians to interpret the testing results and create a process to improve patient care. The next several slides is going to be on a journal article, but it's the psychiatric pharmacogenetic testing, the evidence-based. It was put out on June 2019, and inside this journal, it actually discussed 2 articles from other places that also reinforce the current expert opinion that up to this point, the ability to utilize genetic testing in psychiatry is unfortunately very limited. The evidence supports that only 4 genes have risen to the level of clinically actionable in psychopharmacology. These genes include the cytochrome P450 2D6 or the CYP 2D6, the CYP 2C19, and the HLA-B and the HLA-A that I spoke about earlier. At this time, sadly, that in the practice of the practice of testing in private practice is growing, but really not growing fast enough. It truly is a matter of breaking through the barrier of education. I talked a little bit about cost before, but cost base is actually based on which genes are tested. I did find out recently that pharmacogenetic tests are often covered now by Medicare and in some states Medicaid. Private insurance covers the testing based upon the medical necessity of it. But on the other hand, some laboratories are only offering tests that are reimbursed by insurances, and they're kind of ignoring the value of broader gene testing. Another aspect is kind of reading the reports we're getting. So the reports are getting better and actually are getting a little bit easier to interpret and implement. And this is less intimidating for providers, but in all honesty, again, education is going to be the key growth in this area. The FDA safety communication warning came out against the use of many pharmacogenomic tests, and that was in October. I want to I do want to read you just a little bit of this. On October the 31st, 2018, the FDA published a safety communication warning against the use of many pharmacogenomic tests with a statement. For example, the FDA is aware of genetic tests that claim results can be used to help physicians identify which antidepressant medication would have increased the effectiveness or side effects compared to other antidepressant medications. However, the relationship between DNA variations and the effectiveness of antidepressant medication has never been established. And the FDA is aware that healthcare providers may have made inappropriate changes to a patient's medication based on the result from genetic tests that claim to provide information on the personalized dosage or treatment regimens for some antidepressants. Patients and healthcare providers should not make changes to a patient's medication regimen based on the results from genetic tests and claim to predict a patient's response to specific medications, but are not supported by scientific or clinical evidence to support this use, because doing so may put the patient at risk for potentially serious consequences. That was kind of unfortunate, but the opinion of the author of this journal and of myself, we are just not yet to a point where the panels of testing numerous genes meet those requirements. So we are on our way, though. More information is evolving and will continue with the increase and interest in pharmacogenomics from pharmacists, physicians, and researchers could turn this around and change the way that they feel about that. They had actually had an incident too that they were discussing that about, and I guess that kind of put a bad taste in their mouth. But the psychiatric pharmacogenomics began with the CYP2D6 in the early 1990s. The human cytochrome P450, the CYP, began being delineated then, and TCA's metabolism by CYP2D6 are desipramine and nortriptyline. And these are actually two of the TCAs that were mentioned in the journal, but the FDA-approved pharmacogenetic test first available was the AmpliChip, the CYP450 test. And there's some other tests that are available now, the Genelex and the Genesite. But I just wanted to mention the AmpliChip because it was eventually outdated as the genetic polymorphisms of CYP2D6 and CYP2C19 increased. It determined the phenotype of a patient with two most genetically diverse cytochrome enzymes. Let me go back just a second. Hang on just a minute. I do want to mention this. In the early years of pharmacogenetic testing, the 2D6 could be phenotyped into two different levels, which was an enzyme activity poor metabolizer or an extensive metabolizer. And this could be done by administering a known dose of dextromethorphan to a patient. And then you draw a serum level, and then you take the ratio of the dextromethorphan to the dextrorphan, which was its metabolite, which could be calculated. And then you base the ratio CYP2D6 activity. At that time, you could actually calculate, and it could be determined what that was. So this was one example of a clinical application because it allowed the provider to dose the 2 TCA slowly or aggressively, as we just talked about, depending on that phenotype. Well, we are a little early, but an impressive amount of research and literature has already begun being collected on the increasingly popular topic of pharmacogenomics. And we're just going to conclude with the possibilities of what this research could do for patient care is endless. More knowledge and education will increase the ability to transition this research into usable data, and clinicians should continue to monitor the literature and updated resources to stay ahead of an ever-expanding topic. Does anybody have any questions? Have I thoroughly confused everybody?
I do have a couple questions. So the times when I have used genetic testing, one was that I had a super fast metabolizer for a stimulant, and that's what was my presumption. She seemed to need a lot higher doses, very reliable patient, and this was actually almost 10 years ago, so very early in the process. And the reason I did it was because this patient was willing to pay a separate copay for the second prescription because the insurance would only allow you to get like 60 pills, and she really needed it 3 times a day kind of thing. So I went ahead and I didn't think that seemed fair that she should have to pay. It wasn't a lot of money. She was willing to. We did do the testing, which the insurance fortunately covered. And it turned out that she was a fast metabolizer. And the insurance then did agree to give the 90 per month. So that was one. The other times have been, as you've mentioned, when side effects, new side effects at very low doses. Sometimes, and as you say, when you've got multiple medications going on, you're not getting responses that you might see. Some of the difficulties I've had with it are the different companies trying to push their evaluation systems these days. And I don't really know how to read them to know how good they are. What is of concern to me now, and the one that we're using in our university clinic here, is it doesn't seem to allow you to choose just what you want. As you have mentioned, there's very few that are really relevant for psychiatry, but this one just automatically does things that I don't need. Like, I'm not looking for whether the methylfolate is something that they need to be supplemented with for, say, patients with major depression. So I don't know if this is their cost-effective way of doing it, or is this their way to make money? But it is kind of frustrating because I know it is pretty expensive and only some insurances will cover it. Any thoughts on that?
It is very frustrating. And a lot of the literature that I looked at with the testing, and just from my experience, I think that's just going to take time to kind of cut down on that. I think that they are just putting everything in there. Right now, I don't really have any rhyme or reason why that they're doing that, but I do know that that has been a complaint that I've seen several different times throughout the literatures and the studies that I've read. I don't know that will get any better anytime soon, but that's really all I have on that one. But I have seen that before. You are definitely correct, and you are not the only person that has noticed that. There have been several clinicians.
One quick follow-up question. You mentioned in your presentation that there was D4 for, I believe, ADHD medications. And so I was a little confused. I'm not sure what the D4 portion is, but from whenever I look it up, it appears to be, I think, 2D6, or at least the amphetamines. So maybe you could clarify
that is right. It is 2D6.
But it's a D4 on your slide. Is there some other medications under ADHD, perhaps other things that we use that are not typical stimulants? I mean, we do use things like clonidine and guanfacine, those other ones. I was just wondering if there are some others. I don't typically use those, but I was curious.
Right. Let me see, which slide are you actually referring to?
There was one that just said D4, and I wrote it down because it kind of surprised me if I was maybe not.
Gotcha. It's actually mostly it's the D6, the 2D6 and the 2C19. Those are actually the two that I know of also.
Okay.
Dr. Lawson, I'm curious, has there been any implementation of pharmacogenetic testing at Woodridge on the inpatient side of things?
There's not.
Okay.
Yeah, there hasn't, but I would love to be able to do that. I think to be able to bring that before our PNT committee and to be able to get that started at Woodridge. Unfortunately, I do feel like it probably would have to have a little bit more research and a little bit more validity on that. They would just need a little bit more information before that they would let us start doing that there. I do feel like it's important though, and I feel like it's a good program that would eventually be good there.
But maybe just not yet. Maybe in like 5,
Maybe
10
just
years.
not yet. Yeah.
Oh,
Yeah.
not that long.
Yeah, thank
Yeah.
you.
As we continue to get that research, I think that would be great.
I think the problem, though, is that there seems to be direct marketing to consumers. And so I've actually had patients who have been told by non-physicians, therapists they're seeing, et cetera, oh, you know, you should get genetic testing because they can predict which medication they can use. And these are complicated patients that I've worked with, you know, sometimes treatment-resistant patients. And trying to get them to understand that we don't have that much in psychiatry, particularly that's definitive, and that it is to some degree a waste and an expense. But some are determined. I mean, they're convinced by that. I actually had one patient. There may have been other reasons she chose to, you know, change providers, which is perfectly fine with me. But she really wanted it. We did go ahead. I said, if you want it, you know, we can do, but I don't really anticipate it. And so we did transfer the information to our next provider. But I didn't see anything that came out of it because I really didn't think that was a good reason for it. But I said, well, you know, let's give it a award if insurance covers it. It did not. And that is, I think, kind of frustrating when patients, and you even hear the ads on the public radio. I know, and I think this is correct. I don't want to malign Vanderbilt. They talk about genetic-based testing, maybe for cancer treatment now, not necessarily psychiatry, but regardless, I think it heightens patients' assumptions that there is very targeted kinds of things that really we are on the cusp of or already have, which I think, again, creates some difficulty in the relationship with patients in terms of the reality of what's really out there.
I think you're exactly right. I think that when they put that out there, that it gets patients all hyped up about it's, I mean, it is the next big thing, but I think they think it's a cure-all almost kind of thing. But in reality, you're right, we're very limited right now. And when they come to us, I think that they kind of feel like it's a downer because that we just don't agree with that. But I mean, I think you're right. If you don't feel like it's going to help a patient, you know, there's no reason in ordering something that's not going to be going to help us clinically to increase our patient care. Does anybody else have any questions? I know we're a little bit ahead of time. I'm so sorry. And it's a really tough subject. Like when you start digging in, you can get in deep and over your head. Like I tried really hard to just hit the top surfaces today, but it's a very deep subject, and it's going to be it's going to increase, like it's going to be hard to get all the information that we need over the years. But I think that I think it's going to be a good thing.
Mm-hmm. Cool. Well, thank you very much, Dr. Lawson, and thanks everybody
Thanks.
for attending.
All right. Thank you all.
Thank you.